to finished. And then you'll be free to, we'll have a Q&A session, which Gideon will chair. So my name is Nicole Gordon, and I'm the CEO of Genetics. I'm delighted to welcome you to the latest talk in the Diving into Our Gene Pool series, Genes, Drugs and Jews. And it's all about how our DNA can affect our prescriptions. So a little bit about genetics before we start. Genetics exists to raise awareness, educate, and test the community for Jewish genetic disorders. JGD is a term that we use to describe genetic conditions that are more common in people of Jewish ancestry, including people from Ashkenazi, Sephardi, and Mizrahi backgrounds. Genetics tests for 47 of the most severe recessive JGDs, all of which have no known cure. Since 2014, when we started our screening programs, we have screened over 8,500 individuals in schools, in universities, and through our genetics clinic, educating tens of thousands of young people and identifying numerous carrier couples who have, because of genetics, gone on to have healthy families. We also work with NHS England as the official engagement partner for the Jewish BRCA testing programme, alongside High Cancer Care, which I'm sure many of you have heard of. Since launching the programme, over 32,000 people have signed up to be tested through this groundbreaking programme. BRCA gene faults are at least 6 times more common in those with Jewish ancestry compared to the general population, emphasising the importance of this programme for our community. Both the recessive screening programme and the new Jewish BRCA testing programme give people the knowledge and the power to make the best decisions for themselves and their future and their families' future health. You can find out more information about either of these programs on our website, which is easy, it's genetics.org, or of course you can email the genetics team at any point. The recessive genetics tests we offer are all aimed to have an indirect effect on the individual's screening by helping to prevent children being born with a life-threatening condition. And the BRCA test has a direct impact on the individual by improving the prevention and the diagnosis of certain cancers. Pharmacogenetics is focused on understanding how our unique combination of genes can also impact how we each respond to medical intervention, including how we respond to various medications. This can help doctors make more informed choices about the right drug and dosage for every single one of us. This is the field of pharmacogenetics and the basis for the future of personalised medicine. This evening, we are extremely honoured to have with us Dr. Stuart Scott and Dr. Ben Thomas. In tonight's lecture, we will explore exactly what this topic means, how this affects those of Jewish ancestry, and how the medication and healthcare regulator in the UK are using this knowledge to improve the way they monitor drug regulation. Dr. Scott is a professor in the Department of Pathology at Stanford and a director of the Stanford Medicine Clinical Genomics Laboratory, where he oversees genomic test innovation and implementation. He is certified by the American Board of Medical Genetics and Genomics in both clinical molecular genetics and genomics and clinical cytogenetics and genomics, and his translational research interests include human genomics, pharmacogenomics, cytogenomics, long-read sequencing, and the implementation of genomic medicine. Dr. Scott has published over 160 manuscripts and book chapters on molecular genetics, official genomics, and pharmacogenomics, including its effect in the Jewish populations. At this point, I'm going to pass over to Dr. Scott, who's going to give the first address. Thank you.
Super. Thank you, Nicole, for the introduction. And thanks, Gideon, for the opportunity and invitation to speak today. It's a real pleasure. One thing I did kind of forget to mention, too, is that prior to joining Stanford in 2020, I was at Mount Sinai in New York, and that's where I did my molecular genetics training altogether for about 15 years. So that's in the genetics department there, I was very active in Ashkenazi Jewish screening. And so it's got a lot of my exposure to that kind of population genetics while I was there back in New York. But for today, as mentioned, I'm going to speak about pharmacogenomics and then kind of touch on common themes that you're used to in genetics and population genetics and how that might be relevant for Jewish population. So I'll start with an introduction and then talk a little bit about implementing clinical pharmacogenomics, which is something increasingly institutions and medical centers are wanting and trying to do. And then also end with just a couple of little vignettes about where allele frequencies are unique in the Jewish population. So in terms of pharmacogenomics, you've already kind of heard the background a little bit, but conceptually, it's trying to take a heterogeneous, patient population and understand and parse them out prior to receiving some type of medication. Preemptively, would be the ideal situation. And typically, what we look for is a balance between efficacy, or whether the medication works or not, and the safety or toxicity profile. And so, of course, given those kind of guardrails and outcomes, it would be great if we could identify patients who could either benefit better from an alternative medication or potentially just an alternative dose for a specific medication that already is working. But one of the things about pharmacogenomics that I like to remind people, I think it's important, is that because in the past it kind of got overhyped quite a bit, I think, but it's not every single medication that has an interaction with your genome. It's a much smaller number. And so this is nicely presented in a review now that's 10 years old, but still really informative. Mary Relling from St. Jude's is a real pioneer in the pharmacogenomics space, and looked at all the FDA-approved medications at the time, and did a deep dive into those that were actionable by a pharmacogenomic gene. And at that time, it was about 84 medications out of all 1,200. So what was interesting is when they translated that to the number of prescriptions in the United States, it went from 7% to 18%. And so what that tells us, well, first of all, it's not a trivial, it's non-trivial in terms of the number of prescriptions, but it also tells us that some of these medications are really commonly prescribed. And so it's not just rare disease medications or things like that. So I don't have time to get into all of the molecular genetics and biology around it, but in terms of genes and enzymes, the most commonly studied and reported and tested for when it comes to pharmacogenomics are the drug metabolism pathway genes. And so you'll hear about the cytochrome P450s quite a bit. But these are a very large, diverse superfamily of hemaproteins, and that they're involved in the majority of drug metabolism in the liver and other metabolic reactions, both exogenous and endogenous compounds. But what's, from a genetics perspective, really interesting about them is that they're really highly polymorphic. So meaning that there's a lot of variation in the general population for these genes. And they also have a lot of pseudogenes, meaning copies of them in the genome that are not functional. And so because of that, they're very interesting to study. They can be also very difficult to study because of the architecture of the sequence context. But what we found is that there's a lot of variation in these genes that can actually influence how well you're metabolizing various medications. And so some of the most notable ones are CYP2C9, CYP2C19, and CYP2D6, as these are been shown to be involved with the drug metabolism of about 40% of all medications. So a quick slide on nomenclature too, because I feel like this group is probably well informed when it comes to Mendelian genetics and carrier screening and things like that, and mutation nomenclature. But when it comes to pharmacogenomics, we don't often use the kind of standard nomenclature for molecular genetic testing. What we use is kind of more like the HLA type of testing, which is this star allele system. And so what the system is really supposed to do is tell you the sequence of the whole copy of that gene. So it's really a haplotype-based definition. And so that's sometimes difficult for people to wrap their heads around. But of course, we have two copies of each gene in our genomes, one from our maternal and one from the paternal side. And so when we see a result that says CYP2C19 star 2, that's just telling us, of course, the gene. But then the actual genetic result is the star 2 piece. And so that's telling us that the haplotype in this individual, one of the haplotypes, is defined as star 2. And thankfully, there's a whole group that catalogs all of the star alleles. They're called pharmvar. You can look that up online and quickly find them. And so this is what we use when we do genetic testing for pharmacogenomics and the nomenclature that we report. And then, of course, this is germline genetic testing still. This is not somatic testing or anything like that. And that's just, I think, nicely visually shown here in a small case report from a long time ago for CYP2C9 and acenocumarol sensitivity, which is a blood thinner. And so this was a patient, excuse me, who was very, very sensitive to the medication, only requiring less than 1 milligram per day. And that we know that CYP2C9 is involved in the metabolism of acenocumarol. And so they genotyped the patient and found that he was a compound heterozygous for two loss of function star alleles, star 3 and star 11. And so the nomenclature, when we go to have a genotype, we then infer a phenotype. And so when you have two loss of function haplotypes like this, you're reported as a poor metabolizer. But what's educational about this too, I think, is that if you then, once you look at the family and go back, of course, both parents in this case are heterozygous carriers of each of the two loss of function alleles. The one parent, the father is a star 3 heterozygous, and the mother is a star 11 heterozygote. And so these are classified as intermediate metabolizers. And so star 1 is typically the normal allele, and then the rest of them are usually the variant alleles. And then you can appreciate too that his brother inherited both of the normal star 1 copies, and therefore he is classified as a normal metabolizer. So I mentioned CYP2D6 because if you have any interest at all in pharmacogenomics, you will come across this gene many times probably because it's involved in the metabolism, estimated to be about a quarter of all medications, particularly relevant in the field of psychiatry or primary care, antidepressants, antipsychotics, a lot of pain medications. And even in oncology, there's a lot of literature around tamoxifen response and CYP2D6 status. But what's really helped drive the kind of clinical field forward, and I'll get to that a little bit later, is the availability of actual professional practice guidelines for how to use this information. Because a long time ago, when I was at Mount Sinai, and we set up genetic tests for these genes. Back then, there was no guidelines on how to use the information. So we were so proud and excited of them, but very few people ordered them because the physicians really just didn't know how to use the results. And so, as I mentioned, now we have guidelines to help us through that. And then, in terms of the genetic structure of CYP2D6, it's highly polymorphic. So it's also structurally polymorphic. So there's copy number variants, inversions, duplications, tandem duplications, a lot of interesting germline genetics in this region. And thankfully, the people at pharmvar are doing a great job of cataloging these star alleles. And from a technical side, I'm not sure how much this group is interested in technical pieces, but I'm happy to talk about that offline too, is that from a lab perspective, it's really interesting to try to test for the gene because of the complex structure of the region. It's one of those types of genes where there's all these pseudogenes around it. So to actually get an accurate result is quite technically challenging, excuse me. And then for metabolizer phenotypes, as I mentioned before, we have the typical normal, intermediate, and poor metabolizer categories. CYP2D6 is unique because it also has the gene duplications. So it also has a category called an ultra-rapid metabolizer, in addition to the normal, intermediate, and poor metabolizers. And this has been it's an art and a science in a certain way, but it's really driven by kind of association data from pharmacologists who are comparing, genotype status to like probe drug performance that measures your actual CYP2D6 activity. So these are biological tests. So from a population perspective, though, I think it's really interesting when we look at CYP2D6 and other pharmacogenomic genes to look at how different the frequencies of these alleles are. And so this is a very, very old paper. It's still not really precise in terms of populations and numbers. But going back to CYP2D6, the most clinically relevant phenotype results are the ultra rapid or the poor metabolizers. So those are on the tail of the distribution. And so this figure is just looking at the frequency of those individuals. And so you can appreciate, I hope, that depending on your ancestry, that there's can be significantly different allele frequencies and phenotype frequencies from all around the world. And so this is, of course, fairly limited in its data, but there's a lot of newer papers that have dug very deep into this and reviewed all of the allele frequencies. And it's really, really quite interesting. And some of that's really relevant for what we're talking about today. So in addition to the differences among individuals, we can see actually differences in drug response between ancestry groups. It's not as striking as the difference we see between individuals, but it is definitely detectable. And that's, again, really just driven by these, in part, by the different pharmacogenomic allele frequencies across ancestral and geographical populations. A real clinically important example is carbamazepine, and then the really life-threatening hypersensitivity reaction that can happen in rare individuals. And these have been now identified to be due to specific HLA alleles. And of course, some of these are restricted to the Asian population or the Han Chinese, and then other HLA alleles are far more frequent in the European population. So when I was at Mount Sinai a long time ago, and first getting into pharmacogenomics, that's when we were starting to screen through our Ashkenazi Jewish carrier screening DNA bank, and we identified a few interesting alleles that were more prevalent among the Ashkenazi population that I'll talk about later, but also other alleles that were clinically relevant that were identified in other populations of African ancestry as well as Asian. I think one of the most interesting ones, and this is also a scenario that I'll mention later on the clinical side, but is VCORC1 gene. And this is very important in the warfarin anticoagulant sensitivity. And so this is a single promoter variant, just a G to an A, a simple single nucleotide variant. But it's really strongly associated with warfarin dose requirements. So if you have the G, that's the lower dosing, or sorry, that's the low sensitivity allele, but the A is the high sensitivity allele. And so if you have the A, that means the dose should be much lower and the G is where it should be higher. But if you look at the frequency of the A allele, which is considered kind of the risk allele around the world, if we start in African populations, the A allele is the minor allele with a frequency of less than 10%. But as we go across European countries and populations, then it actually comes to 50-50. And then by the time we get to Asia, it's done a complete inversal or complete inverse where the high sensitivity A allele is now the major allele. And so this is, I'm not a cardiologist, but this is also really quite consistent from what I'm told with warfarin dosing, generally speaking, across different populations from different ancestral and geographical backgrounds. So then, as from a perspective of background, and then how do we use this clinically? Um, that's been an evolving area, and it's not trivial. And there's a whole bunch of pieces that have to come together to actually enable this. But one of the most important ones, as I mentioned before, is the availability of practice guidelines. And so this is a consortium that was started in the States. There's also a European one from the Dutch Pharmacogenomics Working Group. Both of them are very consistent. But the goal for both of these groups is to practice really professional, robust recommendations for clinical practice, but they do not recommend that you should test or you shouldn't test. There's a whole bunch of other people that are arguing about that. The whole philosophy here is that this information is increasingly becoming available. Physicians need to know what to do with the information when they're presented with these type of genetic results. And so, uh, of course, this is all small to read, but I list them here just for awareness, because if anyone's interested or has a clinical question or interest, I highly recommend the CPIC guidelines because they're really structured. They're in the same journal. They become more and more robust over time. They get updated. They have a section on the gene. They have a section on the drug. They have a section on the evidence for the association between the two. Then they have a really clear table on if you have genotype A, here's the recommendation. Genotype B, here's the other recommendation, and also a strength of evidence. So really educational, and it's really helped really push the field forward when we talk about clinical implementation and education and awareness. So we've tried to do some implementation at Mount Sinai and also now here at Stanford. And so we often, from the science perspective or clinical perspective, we feel really confident in it, but we're not always the decision makers when it comes to complete programmatic changes and getting resources for new clinical projects and programs. And so we frequently have to justify this, of course. And so a lot of the rationale for implementing pharmacogenomics seems to be pretty obvious. But like I said before, not every medication has a PGX association. And so we want to make sure we don't over promise on our goals here. But nicely, the European group came out with a really fantastic clinical trial result in the Lancet 2 years ago to show that preemptive testing versus standard of care testing using a 12 gene panel, and I think around a list of about 40 to 50 medications across 7 countries in Europe, showed that it reduced adverse drug reactions by 30% by implementing a pharmacogenomic intervention. So really powerful and really helped confirm what a lot of us were already thinking to be the case. And then the other argument for medical centers to consider is actually healthcare cost savings. So I don't have time to go through all of this, and I'm not an economist either. But this is really hard stuff to do at a complex, large medical center. But at a more focused medical center that also is bundled with the payer side of things, like a Kaiser or a system like that. This is a report from a group in Kentucky that had the ability to monitor what their entire cost structure across the medical center. And what they showed was that after a few years, the cumulative healthcare savings were dramatic after a pharmacogenomic intervention. And that was driven by reduction in emergency room visits, decreased healthcare or decreased inpatient stay, and the third one I'm blanking on, but those top two were really the big drivers. And so this has even prompted some medical centers to just adopt pharmacogenomic testing and pay for it themselves, because the reimbursement landscape is a whole other difficult question that we won't get into today either. But this kind of suggests that there's an argument that even if the medical centers pay for the testing itself, that it could be a benefit for them. So we several years ago we did set up a research program at Mount Sinai that was set up with a 5-gene kind of more focused panel back then, and we genotyped 2,000 patients, and so this is through the Institute for Personalized Medicine there. Erwin Bottinger was the director at the time. And so I was living in the genetic testing laboratory, so we were doing the panel genotyping, but they had invested in a really large IT infrastructure that interacted with our EHR system, which was Epic. And so we would file our results in this database. And then it also had logic that was built in from the CPIC guidelines they were putting into it. And then the nice thing about it was that it was very dynamic and actionable, because when it was set up with Epic, when a physician would go to place an order for one of the medications that was on our PGX list, the system would go look to see if there was a genotype available in the system, and if there was it an actionable result. And so this is an hypothetical example of clopidogrel, which is commonly associated with CYP2C19 loss of function, and individuals have high rate of lack of efficacy and adverse events, or just re-stenosis. And poor metabolizers are actually recommended to get an alternative anticoagulant. And so this was running for several years. And then so it wasn't a clinical trial. We couldn't, measure outcomes. It was really just an implementation study. But a couple of years ago, we were able to look at some data too, just to see provider adoption, because that was the big concern, was that would the providers even care? Maybe they'll just close it out and not even bother. But we were really enthusiastic to see the results from, this is just from one of the examples, simvastatin. And the gene for simvastatin is SLC01B1. And then the frequency of the risk genotypes here, either the heterozygotes or homozygotes for the minor allele, is just under 20%. And so again, many of these fired among the 2,000 patients that were tested. And so this is the pop-up that would occur in Epic, and clearly try to convey to the provider that an alternative statin is recommended. And so we were able to actually track at least what the providers did afterwards. And so again, we were really happy because they were adopting the recommendation almost all the time. And so we had the per CDS adoption is lower because they had already adopted on a previous recommendation. And so the per patient adoption percentage was over 80%. So again, we were really encouraged by that. And just anecdotally too, what we've learned when we talk to providers is that almost all of the time they'll say, yes, this sounds great. I'm happy to do it, but I only have about 7 seconds to spare in my encounter with my patient. So if I have to get another blood draw, or if I have to check insurance, so I have to do all these things, it's gonna be a real challenge. So if you can just give me the information and tell me what to do really quickly, then that's something that has some traction. And so that's this whole concept of preemptive testing and leveraging the electronic healthcare system to help us rapidly implement this type of genotype-driven prescribing. So from implementation now, I just wanted to end off with a little bit on population structure and how it's specifically more relevant to the Jewish population. And so we had reviewed this topic almost 10 years now, back at Mount Sinai too. The citation is here, and it's also been done by others. Basically, I'll just kind of try to summarize that a little bit and use warfarin again as kind of the example of how this can be relevant. And so I think most people know warfarin is another oral anticoagulant. It's been around for decades. But it has a really complicated clinical use. There's wide inter-individual differences in response and a really high dangerous risk of bleeding or stroke. And so that it's considered to have what's called a narrow therapeutic range. And that is monitored by a blood test, a finger stick blood test to measure this, what they call an INR, which is basically just a numerical value for the degree of anticoagulation. And this is what the INR range looks like. If you compare it to safety risks. And so this is what drove the definition of that therapeutic range. Because if you become too anticoagulated, of course, then you have a risk for bleeding. But if you're under anticoagulated, then you have this risk for stroke. And so the goal is to try to get everybody into this range of 2 to 3 and try to keep them safely there. But the problem is that across the general population, the actual dosage that it requires to get people to the same level of anticoagulation is very, very wide in terms of the distribution. So on average, they usually start with about 4 or 5 milligrams a day, but some people are highly resistant and needing up to 20 milligrams, or there's other people who are very, very sensitive and need a much lower dose. And so typically, the cardiologist or physicians will have to titrate the dose to try to get patients into that safe therapeutic range. However, that first few weeks can be a fairly dangerous time for some patients. And so that variability has been known for many years, and it's due to many factors, age, gender, other drug interactions, how much vitamin K is in your diet, but also pharmacogenomics. So we know the pathway of warfarin. So this is a nice educational example, even though now there's some new anticoagulants that have kind of replaced warfarin for a lot of these patients. It's still a really educational pharmacogenomic example, I think. So we won't go through the metabolic pathway of warfarin in great detail, but it's the main point is that it is an active compound and it is metabolized pharmacokinetically by CYP2C9, and that generates these hydroxylated metabolites that are excreted. And it acts on the VCORC1 enzyme. And this is the enzyme that I mentioned before that has that promoter polymorphism that's highly frequent or highly variable in different populations. And then this pathway, by blocking that pathway, that's what it leads to inhibition of the clotting downstream cascade. And so genetic variation in both the pharmacokinetic as well as the pharmacodynamic pathways have been shown to highly influence warfarin dosing. And so this is just an overview of when we look at the different genetic results for both the CYP2C9 gene on the x-axis, as well as the VCORC1 genotypes within these separate phenotypes. And so what you can appreciate is that as individuals have these loss of function star alleles, they have lower dose requirements. But in addition, once they have that sensitive A allele, they also have even more sensitivity to warfarin. But I mention this because there's also a whole other side to this, which is really relevant in terms of the resistance to warfarin side. And so what was found almost 20 years ago now, I guess, was a coding mutation in VCORC1, not the promoter polymorphism, that actually predisposes patients to resistance. And so that's shown here in that these are individuals who required very, very high doses of warfarin. regardless of all of this other genetic variation that I just mentioned. And then it turned out that this variant is highly prevalent and almost specific to the Ashkenazi Jewish population. And the allele frequency of it is around 5%, so certainly not trivial at all. And so, of course, I don't have to explain genetics and population structure in the Jewish population, but of course, it really translates not just from what you're used to in terms of carrier screening, BRCA screening, things like that, but now if you translate that over to these drug metabolism genes, more and more now we have summarized certain allelic variants in these genes that are more common in various different Jewish subpopulations. And so again, here's the VCORC1 example. Oh, sorry. There, thank you. This is the Ashkenazi common VCORC1 warfarin resistance variant. And so again, this is all summarized in the review article that I mentioned before, but in the more recent review article as well, it has some really nice data showing the actual Ashkenazi burden of rare mutations was actually higher in the Ashkenazi compared to other European populations. So again, more and more of these data are coming out with all of the population genomic data that is available for interrogation, kind of confirming what we've already seen in the Mendelian genetics kind of space, where certain Jewish subpopulations have higher frequency of these mutations. On the PGX side, we're finding a parallel story where there's certain drug metabolism genes also carry some of these more frequent PGX variants that have stuck with the population through all the years of bottlenecks and expansions. So I'll stop there and just briefly summarize that pharmacogenomics has the potential to preemptively stratify patients for more precise management based on their germline profile. And I'm really excited about the clinical implementation piece because now we have all these new resources that have increased over the last 10 years, a lot more practice guidelines and other things that the laboratories really require for implementation. And then for this group especially, there's no question that the Jewish populations harbor many unique pharmacogenomic allele frequencies in addition to other variants that are common among populations of European ancestry. So thank you all again. It's a pleasure. I'm happy to, I think there's maybe questions later, but my information is here, my email address, my research lab website. Please reach out anytime directly if I can answer any questions. Thank you very much.
Thank you, Dr. Scott. That was truly fascinating. And if anybody does have any questions that they think they might forget by the end, then by all means put it into the chat. When Gidon shares, he'll pick up the questions. Okay, so without further ado, I'm going to move on to Dr. Ben Thomas. And Dr. Ben Thomas serves as Program Manager at the Medicines and Healthcare Products Regulatory Agency for the Yellow Card Biobank. His research includes dementia home safety in winter, early diagnosis and emergency care studies on COVID and sepsis warning scores. Prior to joining MHRA, Dr. Thomas led the Patient Engagement and Data Research Initiative at Health Data Research UK. Over to you, Dr. Thomas.
Okay, just get myself set up there. Hopefully you can see my screen okay. Good nods there, thank you. And yes, thank you so much for having me in the introductions and for Dr. Scott there and beautifully dovetailing into me. As you can tell from my CV, I'm not a geneticist by trade. I've got quite a varied research background. I've moved slowly into data research and program management. I moved into the Yellow Card Biobank. So thank you, Dr. Scott, there for kind of teeing me up on a lot of points I'm probably going to make anyway. And move my role here today is to tell you more about the Yellow Card Biobank and one of the actions the MHRA as a regulator is doing around the pharmacogenomic space. So hopefully it'll give you some context, some kind of real world applications or research and pilot study that's going on in the UK today. So yes, I've been at the MHRA now for a couple of years, and as a regulator, our job is around drug safety. It's there to protect the UK population in terms of medicines, vaccines, blood products, and medical devices. And we do a lot of this through signal detection, and I'll talk about the yellow card itself in a moment. And the yellow card biobank is a pilot study trying to fit in partly into that space. And looking at a more proactive manner of how we approach things. As a regulator, we're doing drugs, looking at drug discoveries, approval, and monitoring, and part of that is around what adverse drug reactions. Any drug that we take has a reaction, and some of these are much more serious and adverse, i.e., non-intended than we might otherwise expect. I think one of the stats here was already presented last time, but adverse drug reactions are responsible for around or connected to 1 in 6 hospital admissions in the UK. And if we genotype potentially correctly, as mentioned earlier, we can look at a 30% reduction in clinically relevant ADRs, adverse drug reactions. And that's incredibly important for both patients, but also for the healthcare system as a whole, for the impact it has, for the cost it has. And so this is moving into the area that's mentioned around more precision medication, targeting the dosages, getting those correct, can have a really good impact on what we're doing, looking for more designs of new drugs at precision, etc., as we move forwards. And we're interested to support that as a regulator. And in terms of what we'd say pharmacovigilance, I mean, I work within the safety and surveillance part of the MHRA. We're looking to monitor drug safety in particular, and we do a lot around this in terms of informing the public and healthcare professionals around the drug safety announcement and what's going on at any one particular time. We also look at this in terms of the regulatory information and guidance provided. I mean, this is a bit of comparison internationally, but the point about this being that we can provide things around, is a test required, say around a point of care test such as gentamicin and the impact that can have, or perhaps just giving guidance towards that, or just some pharmacogenomic information included on the label to allow people to choose which, or healthcare professional to guide them in terms of medication. As Stuart Scott said, some of that is incredibly challenging to do, and we're at the foothills really of the implementation of that. And what you'll see is that hopefully the biobank can be part of that process and provide a little bit more resources to help clinicians and researchers as they go along this journey. So the Yellow Card Biobank and the Yellow Card is kind of very key to what we're looking at and how we're trying to develop this. It was launched 2 years ago as a pilot, and it's got another year into our 3rd year at the moment. And we're looking here to develop to see how this might fit into improving understanding of how patient genetics influence the safety of medicines. And it's really a test case in some regard for the biobank itself. We're seeing how this can work in practice. We're seeing how we can collect this data, how we can build up this resource, and also do some individual research into particular drug gene reactions as well. But it's not just about that for us. It's about making something which we hope to expand and continue on into the future. I can't talk about what the model's going to be like in the future because in some regard we don't know. This is a pilot, and if you've got thoughts or ideas, yeah, please come to us as well. We're looking to have that input as we go along this process. And why the Yellow Card Biobank? The Yellow Card's very important to us. If you've not heard of Yellow Card before, it's the scheme itself, the Yellow Card scheme has existed since the 1960s, and it gives really the opportunity to hold real-world data on medicines and medical devices that collects data on both of those, and harm that might be happening around those. We've got 1.7 million suspected adverse drug reaction reports on database, and that's forever growing. We had a massive spike around COVID, and you might expect that. And it's a voluntary reporting system where any healthcare professional, patient, carer, industry even can report as well on a potential adverse drug reaction or issue with a medical device that's occurred. And this provides a route for us identifying signals for when there might be an issue occurring. You can do this online, you can do this through an app, and we're trying to increase our ability to do this on a regular basis. You can do this, healthcare professionals, say in your GP practice, on their email system, they will have a little box there saying, yeah, report a Yellow Card if something happens, and we're looking to integrate into NHS app and other things into the future. So it's a really powerful bit of data, but it's not always completely full. We haven't got every issue that might have happened on a reaction. We call this phenotyping the condition and the drug gene reaction. We don't have everything, so more work has to be done. So if you're a patient coming into us for the yellow card process, the first thing you do is you get your prescription, and whether that be a pharmacist or a healthcare professional, wherever that may be. And unfortunately, you might experience some kind of adverse drug reaction to that. And that might be self-identified by a loved one or by a healthcare professional. At that point, you might report a yellow card. And if that happens there, this now kind of brings us into the system that we can do as a regulator for safety and surveillance and get what we call a signal, i.e. we know something's gone wrong, we're going to monitor that and try and do something about it. If it's relevant to something we're looking in the biobank, we will then might invite you to that person to join. And that could either be through directly to the patient if they reported the yellow card themselves. But if they didn't do it, we ask the healthcare professional to contact that person on our behalf and see if they might want to take part. At that point, as long as they've agreed, we consent the patient. We might collect some initial data on them, and then we do at home DNA sampling with them. That then sent to our partner, because we are a collaboration between Genomics England and the MHRA. And we are split in terms of our responsibilities in the work. At the MHRA at the moment, we are doing the consenting, the initial data collection, recruitment, and collecting the sample. But Genomics England are doing the sequencing, the storing of data, and further linking as well to our external data. So anyway, the data is sequenced. It goes to Genomics England into their trusted research environment called the National Genomics Research Library. It's a secure place where researchers can access information. And then we can link that to other health information to give a really good idea of what's going on with that case. So that's data you might have when you engage with the healthcare system. We call it your electronic healthcare records. When you go to a GP, when you go to the hospital, a lot of data is collected. And with permission, we can then link that data in a secure place where it can be accessed anonymously by researchers. And over the long term, we want to keep in contact with researchers, healthcare professionals to let them know what's going on. So what does this look like in practice? Well, we were talking a minute ago about warfarin and the issues around that. And one of the drug genes reactions we've been doing in the past, we've been looking at a couple of them, is around direct oral anticoagulants and severe bleeding, or more commonly known as DOACs. And this is unfortunately does have a known adverse reaction of excess bleeding, and sometimes can be quite severe. And a little story here, we'll call this gentleman Steve, that he was, one day he had a severe chest pain, and he was taken for a CT scan. At that point, they identified several clots on his lung, and he was prescribed DOACs. So a year later on, he unfortunately experienced a severe brain bleed, a stroke. And in his words, he describes an absolute pain in the head, like being hit in the head with a frying pan. Now, thankfully, unfortunately, his body started shaking and closing down. And at that point there, obviously the prescriptions we looked at. Thankfully, he was okay. And as he was reading through the DOAC medication, he saw the yellow card link on that, the yellow card reporting, and he did that. We got in contact with him, and then he joined the biobank. Now, he was very keen to see how his genetics might influence his outcome, and we're hoping to try and support that over time as well. And this is one of our current topics, which we're still collecting participants on, so level around DOACs and excess bleeding. It's what we call an exploratory study, or discovery. We don't know if there is a mechanism of some reaction between genes and adverse drug reaction. There's some potential avenues there, but we don't know for sure, which means we need more participants to be able to understand if there is a direct link. So I've touched on yellow card, and that's very important to us, and we want that to be the main avenue for recruitment, but we've also worked with clinicians. So in terms of DOACs, we've worked with stroke clinics to ask clinicians to identify patients as well. They're very busy. It's a very complicated process to go through for governance and time consuming, but it's a very important avenue for us to get not just to access the patient lists, but also their expertise. And that involves them searching through their patient lists, contacting the patient on their behalf, and seeing if they'd like to be engaging with the biobank. We've also worked with something called the Clinical Practice Research Data Link, or CPRD, who actually exist in the MHRA as well. And they've got a wealth of resources with primary care or GP practices who, it's very hard to get general practice data in this country. It's a real kind of blind spot for us in terms of research data. But CPRD have got a link to, I think it's 30% around that of GP practices who've signed up to help and support research. We've worked with them and they've been a great source for us getting in there. And you might imagine that some of the diagnostic information comes at that primary care level in general practices. But we're also working with patient groups. And that's been a very important source for us, not only for recruitment, but also to start building links, understanding, and buy-in and trust. Any kind of research like this involves having good trust from the public. If we don't have that, it's very hard to maintain this, particularly when it comes around data research. And that was actually part of my previous role in MPEDRI, about patient engagement data research, and this is how we can build and maintain that trust with the public. And it's what we are very proud of so far in this is the trust and buy-in we've had from the public. And it's clear that once engaged, they really do want to take part in this research. People have had this reaction and they want to give back. And I think with the Yellow Card scheme, where unfortunately healthcare professionals, it's been very hard to get reports in. For members of the public, where around 60% of Yellow Card reporters from the public who've reported have joined the study. And if anyone's ever done the research, that is absolutely an incredible number we're very proud of from our public members. And that the progress so far, that strong public engagement and follow through, it's really been a strong thread for us. It also, in terms of our blood collection samples so far, 95% of blood of people who joined the study have had their blood sampled already. It shows how much people want to stay in throughout the process. We've invited over 500 patients, invites sent. Over 90 patients are signed up so far. I mean, that's like an awful lot, but we are a pilot study, but also sometimes you don't need so many people to do this type of research. And also we've had 26 NHS sites signed up to support recruitment to the project. That they'll probably continue into future topics that we'll be looking at. We can't talk about them quite yet, but they are coming in the next year and onwards. We've sequenced over 70% of samples received so far, and that will likely reach close to 100% shortly. So we're doing this, but in the end, we hope to have, hopefully this could be an example for you of what a biobank resource might look like and its use. In the end, what we'll have here is a pool of genetic information and healthcare information all linked together in what we call a trusted research environment. That's where researchers can anonymously access it. And it's going into the National Genomics Research Library, and we are just one part of what's going into the NGRL. Also, what Genomics England are doing, such as the 100,000 Genome Project, means that they're getting a vast group of people in there with a good range of conditions and good range of life experiences. So that means that our cohort within that can be linked easily to that data within that trusted research environment anonymously by researchers as long as they go through an approved access process. For our participants, there's a broad consent which matches with the NGRL's consent so that these people can be recontacted to be included in future research. We don't contact too much, but it's ability to do that. Unlike any biobank, what we're looking for is diversity. We want a good broad spectrum of participants to be in the biobank so it is as impactful as possible. Many things can affect somebody's reaction to drugs. It could be the side of bed you wake on in the morning, your level of exercise, your diet, your lifestyle. But genetics and having a diverse population in ethnicity is incredibly important to us. And at the end of this, we will have hopefully a good, deep wealth of information about each participant and what we call to be well phenotyped. What that means is that we'll confidence, perhaps compared to a more general biobank, that we will know this person will have this condition. And that means that a healthcare professional, an expert, will look at each of the cases and say, yes, they have had that reaction. Yes, they and therefore we can be assured that the research we do on it is more accurate. So what are we looking for? Not looking to going into the future and ways people can help. We want people to help identify and contact patients. When you see anything around the research that we're doing, and that can help us to bolster the supply of participants into the biobank, and that's incredibly useful to us. But we also, if you unfortunately do have an adverse drug reaction, or you know someone who does, please report a yellow card. That helps us as a regulator to be aware of adverse events and to track those over time, and just raise awareness about what we're doing as a whole. At the moment, we've been looking at DOACs and also been looking at allopurinol, which is a gout medication. I don't believe that has any particular overrepresentation in the Jewish community, but future drug-gene reactions we'll look at probably will do. And so I think we'd like to keep this relationship going into the future and keep talking about how we might work together. And that's it for me. Any questions at all?
Thanks, Ben. I'm going to actually just introduce Gidon Schwartz, our Outreach and Education Executive at Genetics. There's a few questions in the chat, and I think, Gidon, you're going to start with these. And I know we've gone to the hour, but if we just take maybe 5-10 minutes for those of you who want to stay in the call.
I'll start with a couple of questions in the chat. Then if people have any more, please ask, and those that have their hands raised, I'll also get to you. So the first one for probably either of you, Dr. Scott or Dr. Thomas, have any pharmacogenetic studies been done covering antibiotics of penicillin and macrolides? And can pharmacogenetics assist with preventing antibiotic resistance in any way?
I can start. So I think there's a little bit of signal from HLA association with penicillins, but I don't think the evidence is to a point where there's anything clinically recommended yet. But I do think there's literature on the research side to show an association there. But I don't know if it's relevant for the resistance question either. Yeah. But the one big one for antibiotics is the aminoglycosides that has the MTRNR mitochondrial association. So those are kids in the NICU, and if they have this mitochondrial variant, they're at very, very high risk for getting ototoxicity and losing their hearing. So that's one that's being clinically implemented.
Great, thank you. I'll pass over to Sarah Levine, who has her hand raised virtually at least.
Hello, hi. So I'm a genetic counsellor, and there's a lab that I work with that's starting to produce some pharmacogenomic results. And I sometimes find some contradictory stuff, and I wonder if you can tell me what you think about this. So sometimes you'll get, for say, the same drug, you were talking a lot about warfarin, and I see this a lot, that you get one gene variant where the result indicates that the patient should have an increased dose, and a different gene variant that says for the same drug give a reduced dose. So how on earth are you meant to interpret that and explain it to patients?
Yeah, I feel bad for you. That's not very good reporting. But it's from the lab side, I should say. But so what's kind of evolved in some of the examples that was showed with the CPIC recommendations and things like that are some of like the easy scenarios where there's a single gene and a single medication, and it's a really direct relationship. In reality, most medications, the metabolism is much more complex with many other enzymes. And so for an example where there's actually two genes, the recommendations should take both of them into account and really should provide a single recommendation. But sometimes there just isn't enough evidence or to actually know how to do that. But the ones where they have done that is on the antidepressants, where there is data for CYP2C19 as well as CYP2D6. So instead of just giving you two different recommendations independently, they put the chart together, and so they map the results to give you a single clinical recommendation based on the results of both genes. But this is, I think, like where the future is going to be going with all the kind of machine learning, more complex combinatorial genetics and phenotype and dosing direction.
So I mean, at the moment, what we can say to people is it's contradictory because we don't have enough evidence, and we'll have to wait and see how it comes out.
I guess, yeah, I don't know which example is it warfarin that you're talking about?
Yeah, I've got one in front of me. I just pulled up while you were talking, where CYP2C9 star 1 star 2 consider reduced dose, and CYP4F2 consider increased dose, but both for warfarin.
Yeah, so the CYP4F2, in my personal opinion, is a much lower effect. So I don't usually even include it when I'm talking about it.
They're over-reporting, effectively.
Pardon me?
It sounds like the lab are over-reporting.
Yeah, a little bit. I would only focus on the 2C9 results that you got. And they should have kind of prioritize those a little bit instead of giving them equal weight, in my opinion.
Thank you. That's so helpful. It gives me a different lens to look through. I think I'm going to feed back to them.
If you want, Sarah, I can connect you with Dr. Scott afterwards.
Thank you.
I think probably the final question from Simon has his hand raised again.
Hi, can you hear me?
Yeah.
Yep.
Yeah, thanks, Gideon. Just a question for Dr. Scott, if I may. Very interested if you could share a little bit about your methodology on the population pharmacogenomics. The reason I ask is our company and myself, we specialize in genetic epidemiology, and we use allele frequency databases like Nomad, you're probably familiar with in the USA, to identify the frequency of causal pathogenic variants. Nomad doesn't have the start allele nomenclature. And to be honest, I've not come across that before. So I just wanted to understand how you get down to that level of detail. Do you start just with the gene and then stratify it using pharma, or how do you get down to that pharmacogenetic?
Very good question. So yeah, it becomes bioinformatically challenging. And so I'm going to put a link into the chat as I'm speaking too. Because in the days when we first started genotyping for some of the SIPs, we would manually do it. You know, we could actually, it's like a Sudoku puzzle, almost like, you know, putting the logic together. But at scale, that's just not possible. So a bunch of different groups have started to put together informatic tools that you can put you can put a VCF file from any kind of NGS output. So as long as it has the appropriate regions in it, like for the CYP genes or for PGX genes, this tool that I just put in the chat, we like it a lot because there are friends who made it, of course, here. But this is the one we're using clinically now in our, we call it our translation workflow, because we have to go from like the variant raw data and now translate it to the star allele haplotype nomenclature. And it can get very challenging. And so thankfully, there's more and more of these tools coming out. And some of them now are even approaching UK Biobank, I think, is one of them, and probably Nomad too, where they want to get that data actually into those databases as well.
Yeah, that's great. I'll definitely have a look at that. Thanks so much.
Yep, my pleasure.
Okay. Well, firstly, a huge thank you to Dr. Scott and Dr. Thomas. I'm sure there's other questions, so I know we can share your email, Dr. Scott, after the event, and we will share the General Biobank email if anybody wants to email directly. And a huge thank you to everybody who has signed up tonight, and watch this space for our next in the series of scientific lectures from genetics. And I would say safe journey home, but that's silly. But thank you all for joining.
Thank you.